At the outset, I would like to thank Probiotic Association of India for giving me an opportunity to present my findings, a part of findings in this informative conference. The title of my presentation today is Protection and Restitution of E. coli and LPS-induced epithelial barrier dysfunction by probiotic lactobacillus fermentum under in vitro environment. Under normal physiological conditions, this epithelial gut barrier integrity is very essential for well-being of an individual. And the essential cells which are maintaining it is a single layer of epithelium connected by these junctions, special structures called tight junctions. Under certain circumstances, due to exposure to pathogen or any toxin, what happens? There are aberrations in the structure of these proteins and led to release of and led to the penetration of these pathogens or toxins within the systemic circulation, leading to different orders. This is a site where the onset and progression of different disorders starts. But luckily for we people, there are certain beneficial microbes, what we call as probiotics. They have shown promising effects to rectify these disintegrated tight junctions. So what we do here, what is our hypothesis in this study is, we used our own isolate, that is probiotic lactobacillus fermentum, to know its effect on epithelial barrier physiology. And we also assessed its mechanism of action. Let me mention here, the probiotic is already characterized to improve the immune-associated barrier disruptions as well as allergy in weaning mice. So what we did, we used an in vitro model. What is often used is Kakadu cells, which resemble very much to the enterocytes. We exposed them to these probiotic lactobacillus fermentum. And what was our experimental design? We have Kakadu cells, we have cells treated with L-fermentum, treated with LPS and E. coli. Then we checked the temporal change in barrier integrity by using transfer assay and by checking the gene expression. Based upon the results from this temporal study, we used different these methods to check the effect of probiotic L-fermentum under these inflamed conditions. The LPS we used is this LPS 026b6, and the E. coli we used as a strain of K12. And what we then did, we checked the immunolocalization of different proteins. We checked the cell morphology by using transmission electron microscopy. We checked barrier integrity as well as gene expression. Before doing the main experiment, what we did, we checked the safety evaluation of probiotic L-fermentum and as well as checked the adhesion index of L-fermentum from different assays, MTT, neutral rat, LDH release. We didn't find any significant change in the metabolic activity of cells, Kakadu cells. Further, when adhesion ability of this strain was estimated, we found 1,850 probiotic cells attaching to 100 these Kakadu cells, which is itself a good significance, which is itself a good adhesion property of the probiotic. The adhesion property was further evaluated by using scanning electron microscopy. Moving to the main experiment, we did transfer assay where we exposed Kakadu cells grown on transfer culture inserts to E. coli, LPS, as well as L-fermentum. And what we observed after 6 hours of incubation with E. coli K12, the phenol red flex was 6.72, and after 24 hours, it was 76.25 with 10 is for 11 CFU of E. coli. With LPS, what we found there was 5.7% flux of phenol red at 6 hours and 31.5 to at 24 hours. But stunning, what we found only 2.45 flux in phenol red permeability at 6 hours and 3.96 after 24 hours of incubation, reflecting that there was breaching of this barrier by E. coli and LPS, but there was maintenance of integrity by this probiotic. We then use a different combination of probiotic, that is displacement assay, competition assay, and exclusion assay. In displacement assay, the cells were pretreated with probiotic. In competition, both were given together. And in exclusion assay, there was pre-exposure of pathogenic LPS as well as E. coli K12. And what we found a significant decrease in the flux of phenol red when cells were treated with L-fomentum under displacement assay, competition assay, as well as under exclusion assay. Similarly, with LPS inflamed cells, there was a significant reduction in the transvectorial this flux of this phenol red in displacement, competition, as well as in exclusion assay. To prove it more, what is the effect? We use immunofluorescence microscopy. These are cells exposed to E. coli. These are cells to LPS control and L-fomentum. You can see there is complete loss, disrupted and disorganized actin filaments in E. coli as well as in LPS. But when we compare these two cells, there's a mesh of actin filaments spreading throughout the cell. Similarly, we do it for Claudin-1 protein, which is an important tight junction protein. The similar results we find there. There are contrast boundaries of the cells in control as well as L-fomentum treated cells, but there was a significant reduction in the intensity fluorescence with E. coli and LPS. Similarly, we do it for Z01 protein, which is an adapted protein. There was decrease in the fluorescence, but there was cells with perfect morphology under control and L-fomentum treatment. We then use exclusion assay, competition assay, displacement assays. Again, these are cells treated with L-fomentum and E. coli, and these are with LPS and this L-fomentum. So what do we see here? It was in E. coli there was loss, in LPS there is loss in the intensity, but here they are reverting back to their original, almost original conformations. Similarly, in case of Z01, Claudin-1 staining, there was increase in the sharpness of the boundaries present within the CACO2 cells. Similarly, when we do for Z01 protein, the cells exposed to this E. coli as well as LPS, they were trying to restore their original conformations. To validate further what we did, we did transmission electron microscopy. These are control cells, probiotic treated cells, and E. coli treated cell. What happened? There is this increase in the paracellular transport. What is happening? There is increase in this intercellular space. What is happening in most of the disorders of intestines? We then used same methodology, exclusion, competition, and displacement, and what we found, the cells treated with probiotics in different combinations were retaining their conformation back to the controls. So we check next, we asked a question to ourselves, what is the response of the genes associated with these barrier functions? We used gene Z01, Claudin-1, occludin, cingulin, and PIGR and beta-defensin. Let me mention here, PIGR and beta-defensins are immune-associated genes, and these are barrier-forming genes. So what is happening here? There is decrease in the expression of all these barrier genes after exposure to E. coli K12, but there was an increase in the expression of PIGR and beta-defensin. Similarly, with LPS, there was decrease in the expression after 6 hours of incubation. However, when cells were treated with L-fomentum, we have a contrasting results. There was a significant increase in the expression of main tight junction forming proteins, that is Z01 and all these, but there was an increase in this expression of PIGR as well as beta-defensin. But it was not as significant as we have seen with the E. coli treatment. Then we used different combinations like we did there. There was significant increase in the expression of genes when cells inflamed with LPS or E. coli were treated with probiotic. L-fomentum in different combinations, exclusion, we have competition, and we have displacement. So what to conclude? Probiotic L-fomentum attenuated E. coli or LPS-induced change in epithelial barrier functions. And let me conclude with the saying of Hippocrates, let food be our medicine. This is the best possible choice. Thank you very much. I would like to acknowledge DBT for providing the funds.
Quite in depth, very nice. You when you were looking at three different types of experiments, you were calling them differently. One is exclusion, whatever. So what I understand is one you pre-treat
Yes, sir.
with lactobacillus. In the other one, together, and in the third one, you treat after the insult has taken place.
Yes,
Is
sure.
that correct? And in all three, you find identical results.
Not identical.
Almost.
In this displacement assay, the results were not as good as were shown in pre and competition treatments.
And how would you explain that after the injury, after the tight junctions have been disrupted by E. coli, and that too you use K12, and K12 is inducing that amount